Hello everyone, this is part 1, so I'm going to be answering from question 1 to question 4 for the Unit 2 October 2024 past paper for Pearson Edexcel. Let's get started. Okay, so the first question shows an incomplete plant cell. Okay, so hopefully when you look at this, you remember that this is called a root hair cell. So this type of cell is found in plant roots. Okay, so label the tonoplast on the diagram and then draw and label a nucleolus in this plant cell. Okay, so over here is our tonoplast. This is our tonoplast. This whole area is the tonoplast. I just drew the line in the middle, so I'm precise and accurate. And then over here we have the nucleus, and the nucleolus is found inside of the nucleus. Make sure when you're labeling, just draw a straight neat line. Do not do an arrow. So if you do an arrow like this is incorrect. If you do a simple straight line, this is correct. Okay. Complete the table to show which of these structures would be present or absent in plant leaf cells. Okay, well, a myeloplast. A myeloplast is responsible for starch biosynthesis and storage, so it is present, okay? Chloroplast, of course, is very famous, has chlorophyll, okay, for photosynthesis, is present. The Golgi apparatus is used for modification, so it is also present in plant cells. Plasmodesmata is also useful for connections to transport mineral ion between different cells. Therefore, it is also in plant cells. Okay, the photograph shows a lysosome. So just remember that a lysosome is simply a bilayer. It's basically a bilayer that holds in the middle enzymes. So we just have our lipid bilayer that is all around in a circle. So we have our phospholipid heads. We have our protein heads and our phospholipid tails. And it just looks like that all around. Okay. Calculate the actual length, the actual width, from X to Y of the lysosome in micrometers. Okay, so the magnification is times 250,000, and I measured the distance from X to Y in millimeters, which gave me 30 millimeters, or it's going to give you 3 centimeters, and then you convert it to millimeters. So I'm going to use this triangle over here, the I-am triangle, where I have I as my image size, A as my actual, M as my magnification. So to find the actual, it's going to be dividing the image divided by the magnification, both units in micrometers, which is going to give me 0.12 micrometers. To convert millimeters to micrometers, all you do is multiply by 1,000. Okay, question number 2. Diatoms are single-celled organisms important for many ocean food chains. The photograph shows a diatom as seen using a scanning electron microscope. Okay, diatoms contain a nucleus, mitochondria, and chloroplasts. All these are characteristics and features of eukaryotic cell. State the domain which contains diatoms. Okay, so since these are features in a eukaryotic cell, therefore the domain is going to be eukarya. Okay, the nucleus in a diatom contains DNA. Name 2 processes that take place in the nucleus of a diatom. Okay, so first of all, DNA replication. You can also mention semi-conservative replication, which is the same thing. And you can also, for the second point, mention production of mRNA, which is called transcription. Remember, you cannot mention translation because translation does not occur in the nucleus. Translation occurs in the cytoplasm. Okay, it is predicted that the diatom populations will decrease in number due to global warming. Scientists have suggested that this will cause a decrease in ocean biodiversity. Describe how a decrease in biodiversity within an ocean habitat could be determined. Okay, so the first point is when you're talking about a range of species, okay, and you're going to do a study in the ocean. The first thing that you want to determine is determine the species richness. Determining the species richness means just finding how many different species there are present in the habitat that you are working on. Okay, and then within these species, you need to count how many individuals are in each species. So the second point is going to be then count the number of individuals in each species, okay? And then you're going to calculate a diversity index to find out the amount of diversity that we have. Okay, question number 3. The photograph shows a Javanese Edelweiss plant. This plant is endemic to Indonesia. State what is meant by the term endemic. This is one of the words that you need to memorize for your exams. So how I memorized it was very simple. Remember the pandemic, COVID-19, the pandemic, the COVID was spread all around the world, okay? So pandemic all around, endemic only in one place, okay? So therefore, the plant is only found in one geographical location, okay? A student used a microscope and a graticule to view a pollen grain from this plant. Over here we have our eyepiece unit, and we have our eyepiece units over here. When you zoom in, or you have the physical paper, you can count each part. However, from 30 to 40 it's 10, from 40 to 50 is also 10, therefore it's 20, and then we have 2 on one side and 1 on the other side, so our total eyepiece units is 23. Then we're going to have our equation, which is length in millimeters divided by the eyepiece units is going to be equal to 0.1 millimeters over 100. So how did I get this? I got it from over here, where it said 100 eyepiece graticule divisions is 0.1 millimeters. Okay, so we're gonna be looking for the length. Okay, so length is going to be on top of the 23 because 23 is the eyepiece units that we calculated. We're going to move this 23 by multiplication over to the other side, which is going to give us 0.023 millimeters. Now we need to convert this to micrometers because this is our main option over here is micrometers, and it's not 2.3 millimeters. Okay, so to convert to micrometers, all you're going to have to do is multiply the millimeters by 1000, which gives you 23 micrometers. Now state what is meant by the term magnification. Okay, so magnification is how many times bigger an object appears in an image than it is in real life. Okay, because you zoom in on an object, you make it look bigger when you're using the microscope. Okay, the diagram shows a germinating pollen grain. Describe the roles of the tube nucleus and the generative nucleus. Okay, so this question is very important and it's repeated in multiple exams. So create a flashcard of this so that you can memorize the steps and each function of the tube nucleus and the generative nucleus. Okay, so the tube nucleus is over here on the tip. Now, since it's on the tip over here, it's only going to make sense that it's going to release enzymes. Therefore, it releases enzymes and it controls the growth of the pollen. So the first point is it controls the growth of the pollen tube. Secondly, it controls the production of hydrolytic enzymes. Remember, these hydrolytic enzymes, they break down the tissue in this tile to pave the way for the pollen tube to grow. Now, the generative nucleus divides to form 2 haploid nuclei. Now remember, one nucleus is going to fertilize the egg, and the other one is going to fuse with the polar nuclei and create the endosperm. Okay, question number 4. The photograph shows a sperm cell beginning to enter an egg cell. Okay, you can see here some form of, penetrating action over this jelly-like layer, okay? And the acrosome is probably located over here. So a sperm cell swims by rotating its flagellum. Which of the following statements about sperm cells are correct? Number 1, sperm cells are diploid. That's incorrect. The cell is going to become diploid when it's fertilized. sperm cell alone are haploid. The flagellum rotates using energy. Yes, it rotates using energy because there's going to be mitochondria located over here, which through respiration is going to produce ATP for the cell. Sperm cells have a streamlined shape to increase resistance. No, wrong. Therefore, it's going to only be 2 only. If it's going to be streamlined, it's going to be this shape. Okay. However, the sperm cell is not this shape. The sperm cell is more like a circle with a box over here and a tail. This position is not streamlined. This is streamlined. Describe how the nucleus of a sperm cell enters an egg cell. Okay, so over here in this diagram, you can see that there's an acrosome, like a sac on top of the head. This acrosome releases enzymes, okay? Therefore, the enzymes are gonna digest the zona pellucidea, which allows the sperm cell to fuse with the egg cell. Over here are 3 points, therefore you're gonna get the 3 marks. The cortical granules in the egg cell contain enzymes. These enzymes prevent more than one sperm entering the cell. Okay, this is a repeated question in many past papers, so also create a flashcard of this. So describe the role of the rough endoplasmic reticulum and the Golgi apparatus in the production of these enzymes. Okay, so first of all, translation is going to occur on the rough endoplasmic reticulum, because the rough endoplasmic reticulum contains ribosomes. These ribosomes are needed for polypeptide synthesis. The formation of tertiary structures also occur on the rough endoplasmic reticulum. Remember that proteins are going to have different structures. We have the primary, the secondary, the tertiary, and the quaternary. Then the protein is going to be modified in the Golgi apparatus. Okay, suggest how these enzymes will prevent more than one sperm nucleus entering the cell. This is also a repeated question. So the cortical granules, as you can see over here, okay, will fuse with the cell membrane and release enzymes. These enzymes will thicken the zona pellucidea. Over here you can see the zona pellucidea, so it becomes much more thicker and stronger, meaning it's harder for the sperm cells to penetrate through. This means that the acrosome can no longer digest the zona pellucidea. Therefore, the sperm cell is not going to be able to enter the cell. Thank you so much for listening to part 1, and please subscribe and comment if you need part 2. Thank you so much for listening, and I hope that this was helpful.